Hello everybody, today we are going to study about cell, the fundamental unit of life. The cell is the smallest or basic unit of structure and function in living beings. It is being called the fundamental unit of life. Why it is being called as fundamental unit of life? Because this cell, all the body is made up of a single cell. So we could say a cell is the structural and functional unit of life. Why we are saying this? That cell is the structure and functional unit of life because the body structure is being also made up of cell and the functions of the body are performed in the cell also. So we are saying cell is the structural and functional unit of life. Now coming to the study of cell. The study of study of cell structure is known as cytology. Cyto means cell and logy means study, to study. So the study of cell structure is known as cytology. In cytology we mainly deal with the structure, only the structure and the morphology of the cell. But it is being now modified into cell biology. What is cell biology? In cell biology we deal with the cell physiology of cell, reproduction of the cell, reproduction of cell, and cell chemistry. That means the reactions taking place in the cell. Now coming to the, I hope you have understood the cytology, difference between cytology and cell biology. So I am going to tell you again the difference between cytology and cell biology is that cytology is the study of cell structure, that is the outer portion, the morphology of the cell. But now the cytology is being broadly or extended into cell biology, which deals with physiology of cell, reproduction of the cell, and the internal structure of the cell. Now coming to the discovery of the cell, who has discovered cell first? Cell was first discovered by person named Robert Hooke. Where he has observed the cork cells. Cork cells are present in the bark of a tree. Observed cork cells of bark. These cork cells are dead cells. Dead. So, the cells discovered by Robert Hooke are dead. He observed that the cell looks like the honeycomb-like structure. Honeycomb-like structure. He said that the cells he observed looks like honeycomb-like structure, and he said these small chambers he named as cellulae, which means small rooms. From this word cellulae, he has given the word cell. Robert Hooke has discovered dead cell. Now coming to the living cell, who has discovered the living cell? The living cell was discovered by Antony van Leeuwenhoek, a scientist named Anton van Leeuwenhoek observed or reported first living cell. He is also being known as the father of microbiology, Antony van Leeuwenhoek. He has observed the first living cell, that was a bacteria. So if a question arises who has discovered cell, then you have to answer Robert Hooke. But the first living cell was discovered by Anton van Leeuwenhoek, and that was a bacteria, the prokaryotic organism. Robert Hooke has also written about his discovery in his book. What was the name of his book? Micrographia. In this book he explained how he has discovered cell. Now coming to the cell theory. The cell theory was first given by two scientists named Sheldon and Schwann. Schwann. What they said? They said that cell is the basic unit of life and all organisms are made up of cells. This was a theory given by Sheldon and Schwann, and later on this theory was modernized, elaborated by Rudolf Virchow, which is also known as cell principle, cell doctrine, and also modern cell theory. So what he had said, it was given by, as I already told you, Rudolf Virchow, a scientist named Rudolf Virchow. The main 3 points of the modern cell theory are that all plants and animals are composed of cells. Second point what he gave, cell is the basic unit of life. Cell is basic unit of life. And the third, the most important one is that all cells arise from pre-existing cells. Omnis cellula e cellula. Omnis cellula e cellula. The main point of the modern theory was that all cells arise from pre-existing cells. So the cell theory is applicable on all the living cells, but there is an exception to cell theory. What is the exception of cell theory? Is that virus. Virus, exception of cell theory. Why? Because virus comes between the living and the non-living. Non-living and living. It get activated when it enters inside the body of a host. It cannot work independently because it is not having its own metabolic machinery. So virus is exception of cell theory. Now we'll study about, we'll are going to know how the cells are being observed. So today we are moving to microscope. Microscope. Microscope is an instrument that is used to visualize the objects or organisms which are not visible to us by the naked eye. So we'll expand this word first. It is a Greek word. Microscope is taken from a Greek word in which micros means small and scopion, it means to see. So microscope is an instrument used to see objects that are not visible with the naked eye. Now we have already told that I have already told you that the first cell was discovered by Robert Hooke. So it's natural the first microscope would be discovered by Robert Hooke. The microscopes we are using in the laboratories are the compound microscope. Compound microscope, and they are also known as light microscope or light microscope. These compound microscope and light microscopes were first invented by Janssen and Janssen. And Janssen. Why it is being always a light microscope? As it's natural, light, it is being using light as the source to view the specimen. Then another type of microscopes are the electron microscope. They are very advanced type of microscope. Electron microscope. These electron microscope were discovered or they are invented by Knoll and Rashka, two scientists named Knoll and Rashka in about 1931. And this microscope is used in research purposes. Research. So what the microscope which we are dealing in our schools is the compound microscope. Compound microscope which was discovered by Janssen and Janssen. Just coming to the structure of a microscope and how it works. A microscope has a eyepiece and here it extends like a tube. At the end are the objective lenses. These being known as the eyepiece from where we visualize. And these are the objective lenses that increases the magnifying power. Here is placed the stage where specimen is being placed. Here is the tube for holding the microscope. Here it's just a rough diagram. Here is the mirror for focusing the light on the specimen. So we keep the specimen over here, this place, and we keep our eyes on this. With the help of these these lenses, this microscopic organism or whatever the object we are taking would look little bigger, and we'll be able to see its internal and its morphology with the light microscope. How a specimen is prepared, just have a look how a specimen is prepared. If for example we take onion peel example. Onion peel. For onion peel we have to take a onion and cut it. From the inner side you will see a thin membrane like structure, and we'll take out the thin membrane like structure and put it in a watch glass. So watch glass with water, inside it is water, so that it doesn't get dry. We will take that specimen onto the slide. This is the specimen. We'll put saffronin. Saffronin is the stain, red color, that will provide red color to the specimen. After putting the saffronin, we'll put a cover slip on it. We'll put a cover slip over the specimen. When we put the cover slip over the specimen, the extra water or the extra stain comes out, and now it is ready to be visualized under the microscope. There are a number of stains we can use, but in our schools we generally use saffronin. Other stains like methylene blue and crystal violet are there that are used for higher purposes. Now coming to the types of cell. There are mainly two types of cell. Types of cell. There are two types of cell, prokaryotic and eukaryotic. What these terms mean, first of all look at that. Pro means before and karyon means nucleus. So here in prokaryote the nucleus is very primitive, but in you means here the nucleus is true. U means true and karyon means the nucleus. In eukaryotic organisms the nucleus is well developed. Now coming to the difference between the prokaryotic and eukaryotic, we'll just have a look at the main basic differences between the prokaryotic and the eukaryotic cell. In prokaryotic, as I already told you, the nucleus is primitive, that is incipient nucleus. No nuclear membrane. No nucleolus. But in eukaryotic, both these all are present. In eukaryotic, there is a well-developed nucleus, and here it have a nuclear membrane that is also a double membrane that you will later on study when I'll be taking the nucleus in detail, and it has a nucleolus also. Coming to the DNA of prokaryotes, it's circular DNA in prokaryotes. But in eukaryotes, there is a linear DNA. In the next class, we will dealing with the other differences of the prokaryotes and the eukaryotes.